right, Hans. That's really, really great to have you.
Yeah,
You know,
thank you for having me.
I was really, really excited for today because you are using enzymes, right?
Hmm.
I grew up doing biocatalysis in, well, back when the dinosaurs were walking on Earth, to be honest. So, but I did biocatalysis during my PhD. That was when directed evolution was just starting.
Hmm.
I followed the field, you know, from the distance, if you wish,
Yeah.
in many years, and so it's really nice to see what you can do with that now. It's super exciting. So I can't wait to go into it. But so as I said, we're going to have this sort of relaxed conversation, and what we are really interested is the relationship between the person and the science, because
Okay.
very often that gets lost a little bit
Hmm.
because you're a name on a paper, right? But there is a strong influence we have seen doing this podcast between the personalities and what you actually do, and so that you will see that I will go on the personal side a little bit,
Hmm.
but so if you don't mind, I start from reading a very brief summary of your professional descriptions, your resume. So you tell me if there's anything wrong, um, and then we kick off. Okay.
Okay.
So you are currently associate professor at Script Research in Florida
Yes.
at the Department of Chemistry.
Hmm.
You did your undergraduate work at Columbia University
Yes.
when you worked with Gilbert Stork and Ronald Breslow.
No, that's not
Uh,
correct.
it's not. Oh,
No, no,
what?
no. I worked for Ronald Breslow for about a year, and then I switched group to work for Kristen Lambert, who was just starting out as
Oh,
an assistant
okay.
professor at that time.
Thanks.
Yeah.
Not taken. Perfect. Okay. And then after your PhD, then you actually joined Scripps, right? For some graduate work, and
After
that was
my
with in the Phil Barron group, is
Yeah,
it?
I did my PhD with Phil Barron.
You did your PhD there, yeah.
Hmm.
And then you did a postdoc with Francis Arnold,
Correct.
Nobel Prize.
Hmm.
Was this before the Nobel Prize?
I joined before she got the Nobel Prize.
Okay, that must have been amazing
Yeah.
time. And then back to Scripps for your current position,
Hmm.
is it?
Correct.
Okay, perfect. Thanks. All right. So Hans, great to have you with us today. And as we always do, we like to start from the beginning, right?
Yeah.
To the kind of reconstructing a little bit the path to your current role and how you
Mhm.
develop your scientific interests.
Yeah.
But what I'm really curious of is, how did you even get interested in science in the first place?
Uh, well, grew up in Asian society, and I feel like there's always a lot of pressure when you're growing up to be either an engineer or a doctor. And I guess from an early age, I've always been drawn to math and science. Um, and yeah, I think it just went from there. I so I guess a little bit of a background. I When I was 15, I was awarded a scholarship to study in Singapore, and so I spent 4 years there. And in the Singapore education system, there's the O level and the A level. It's like the British system. And
Yeah.
For my A level, I went to this school called Raffles Junior College, and uh, they really have a very strong chemistry program. So every year they would send people as part of the Singapore chemistry Olympiad team. So I think that was also how I got drawn into studying chemistry. It was that, I guess a little bit of a motivation, or just trying to be competitive, to see how far I can push myself. And along the way, I fell in love with the subject.
And then, after you completed your early studies, then you went for another international relocation, and you went to the US to join Columbia
Mm-hmm.
University. So quite a lot of international experiences for you at a young age.
Yeah, there was a lot of traveling, getting exposed to different cultures. Yeah.
Can
It
you
was,
comment on the challenges? How was it for you?
well, I think the biggest challenge I faced was when I first moved to Singapore. Uh, I was on my own. There was part of a scholarship, so I stayed in a boarding school. So the first few months were hard. I had to learn to be on my own, away from my family. So I was homesick for a little bit, and also the challenge of um, picking up the language. I guess English wasn't my first language, and in Singapore, their national language is English. Uh, so just getting adjusted to communicating in English is also another challenge. Yeah, but I think I got used to it after about half a year, and was much better after I got adjusted.
So was it going to the US easier compared to
Uh,
going to
yeah,
Indonesia
I would say
and
so.
Singapore?
I would say so. At least I was already used to living on my own, away from my family, and uh, what's nice is that when I got to Colombia, I knew a few people from my high school that also went there, so I think it helped with the adjustment, just having that small circle of friends that you can hang out with and stuff.
So do you think that your experience at a young age,
Mhm.
leaving the family still very young and going abroad for school and then going to another country for university, being
Uh-huh.
prestigious organization like Columbia, has it given something that some other people with a sort of easier upbringing might not have? Do you think this has given you an edge as a person or
I
as a
would say
scientist?
I don't know if it really gives me an edge, but I did learn a lot about being independent and just being able to look for uh, something to motivate me if I'm down. Uh, yeah, I think in that sense it helps with when I'm faced with challenges, and then.
Makes sense. So
Mhm.
that from the character viewpoint, made you more resilient, maybe. Isn't it?
Yeah, I guess so. I think that's true, and I think the other thing is that uh, in those 4 years, I spent them in a boarding school. So there's like this structure that's in place for you, and I think that helps with just being more disciplined in your day-to-day work.
So you feel like the experience is still with you somehow. So you're the person you are because of what you've been through, as in
Yeah,
your education.
I would say so. Yeah, yeah,
It's interesting and it's precious. And many people hate it, right? So many people,
yeah.
you put them in a boarding school, you put them around structures, and they
Oh, I
take it
mean,
in the
yeah, I
wrong
mean,
way.
I wouldn't say there are times when I don't like it. That I remember that we have a set time for like a set study time in the afternoon and in the evening, and sometimes you just didn't want to go to those sessions. It's just I think part of our nature, especially as
Yes,
teenagers.
of course,
Yeah,
of course, I can imagine. But being disciplined when you're older is actually very helpful, right?
yeah, yeah, for
and,
sure.
and in science, I can see how that can help.
Yeah, especially in this line of
So
work.
yeah, so as you progress with your chemical studies,
Hmm.
you started as a pure organic chemist, I would say,
Correct.
right? So
Hmm.
you've been focusing on some of the most complex synthetic targets that seem to be very exciting for organic chemists.
Yeah.
Doing these sort of natural compounds that are incredibly complicated, and
Hmm.
where you need to design the synthesis for,
Yeah.
with 40, 50 steps, and
Hmm.
um, these these are an incredibly strong exercise, right, to develop creative chemistry and,
Yeah.
and looking at it from a certain perspective.
Hmm.
So How 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 did you like that? How did you approach that?
I still love doing multi-step chemical synthesis, and I of course love reading those on publications. I think what I really enjoy about the process is this deconstruction process of trying to dissect a very complex molecule into uh, simpler and simpler fragments in a, I guess we call it retrosynthesis, where you try to work backwards from the target molecule to simpler and simpler building blocks. I think I really enjoy that thought process of trying to uh, disassemble things in a reverse sense and then trying to assemble it back. And and along the way, I think there's a lot of challenges that require a lot of problem solving skills. And I think that was always really fun, just trying to troubleshoot reaction. Like, say something doesn't work, but you know that it's it works somewhat, it gave low yield, and then you try to figure out why it's not working. And then just trying to figure out how to optimize the conditions. I think for me is very rewarding. Just the entire process.
The way I've looked at I've always looked at this type of work is obviously I can see the chemistry angle, how exciting it could be, and as the way you describe in deconstructing the molecule and trying to find a way to get there.
Yeah.
But in a way, you have nature that does it so beautifully, and it's
Right.
really, really hard to compete, isn't it? So have you ever, because very often the approach the chemists find is remarkably different from what nature does, right?
Yep.
So do you always or ever get inspired by how nature does it? How what was the the bio pathways to those molecules, and what can we learn? And maybe I'm going towards the use of enzymes somehow,
Yeah.
as you can imagine. But obviously, nature runs reactions in a very different way, but it also runs in a very different environment, right?
Yes.
Doing it in the cell or doing it in vitro is a completely different story.
Yeah, I think that there's a subset of synthesis where we try to mimic how nature does the transformation. I guess we call it biomimetic chemistry, where we try to emulate what nature does in the lab. And I think you can learn a lot of things from here. You can validate the pathway that someone proposed for the biosynthesis of certain molecules, and I think you will also learn lessons about chemical reactivity that way. For my PhD work, I think I would also say that part of what we were trying to do was inspired by how nature does things. So specifically, we wanted to do this exercise whether we can start with a carbocyclic skeleton that contains no functional groups on it, and we try to see if we can put on functional group, in this case hydroxyl groups or alcohol, in an efficient fashion to get to the target that we want. And we did a proof of concept with steroids. We had a particular steroid target in mind that's heavily hydroxylated, and we wanted to see if we can achieve this oxidation sequence through chemical means, purely chemical means. So I think in that sense, we were trying to emulate nature. It's just that the toolbox that we have is completely different.
How do you do that chemically?
So we would have to rely on existing functional groups on the structure itself. So it's not completely unoxidized, and so there's like one or two existing functional groups on the molecule that we can use, and typically we would use this to direct reactivity at different positions to introduce the additional functional groups. And in our manuscript, we label, or we term this idea as a relay strategy, because we're relaying oxidation state, if you will, from one position to another.
I understand.
Mhm.
And so gradually you evolved, I suppose, your toolbox. And when was it that you first encountered the enzymes?
That was, I think, started considering enzymes maybe towards the end of my PhD. I think at that point I had some time to reflect on what I have done, and I realized that I was able to make the target that I wanted to make, but that took 20 steps. And I realized that some of the transformations that we were using to introduce some of those hydroxyl groups that we wanted are rather inefficient. So, for example, we needed 3 steps to install one of the alcohols, and I thought that would it be nice if we can do this in a single step? But we realized that the tool is just not there with for doing it with purely chemical means, at least at that point. So I began looking for potential solutions, and that's when I came across some of the work that's been done in the enzyme world, and that's when I start considering working with enzymes.
So oxidative enzymes are one of the more difficult parts of bio, and probably something that still needs quite a lot of exploration, isn't it? So I suppose you were using something like cytochrome P450 or some other families of these enzymes.
Correct.
So and I can see how this at some point brought you to Francis Arnold. So I guess you were looking at improving your toolbox and see how you could make the best out of biocatalysis, how the expert did it.
Correct. Yeah, that's correct.
So obviously that is what now characterizes your work. So the uniqueness is in the way, as an organic chemist, you have embraced a tool which is probably fair to say underutilized by the community at the moment.
Yeah, but it's growing definitely, right?
It is. Why do you think? Because biocatalysis is not new, and to be honest, commercial applications go back to many years, right? And there are some brilliant examples, in particular in pharma, but also in other industries, right, where enzymatic steps in synthesis play a key role.
Yes.
So the value is there, and it is evident. But yet, at least in my years, I've always seen traditional organic chemists a bit skeptical about it, or maybe it's because they are not familiar with it. I don't know. What is the reason in your view?
I can think of a few things. Just one thing that first came to mind is that when you look at the applications in the pharma industry, a lot of it involve the use of lipases, ketoreductases, or imine reductases, or transaminases. And in some ways, I think that these are probably the more routine transformations that you could do in some ways with traditional chemical methods. It's just that the enzymes do it a lot better in terms of getting the right selectivity and so on. So I think that was one of the reasons why people did not really consider using enzymes too much, especially in academia. And I think
for you, the beauty is opening up the possibility to run completely different reactions, right?
Right. Yeah. So I guess in my independent work, I wanted to address current gaps in the existing toolbox that organic chemists use in their day-to-day operation. That's why the work in my lab focuses a lot on the use of these oxidative enzymes.
So you're currently focusing on oxidative enzymes because they give you something that you can't really do it with alternative methods.
Right.
Is there any other enzyme class that you're interested in, even though you might not use it yet? Or is there any other high potential area that you see?
Enzymes that can form carbon-carbon bonds will definitely be interested in. Thought about it every now and then. It's just that we haven't had too much time to work on it. I think in general, yeah, I think that's probably the one thing that uh, intrigues me the most at this point.
Yeah, I can see, I can see why.
Especially if you can, you know, if you can stitch together different fragments of carbon-containing fragments at interesting positions, I think that would be awesome.
Yeah, indeed, of course. Uh, do, do you find it complicated from the sort of operational perspective? So if I think about how a traditional, synthetic organic chemistry rely on more chemical methods work, you can have some sort of transition metal catalysis type of methods, which is relatively easy to set up, right? You have relatively low number of ligand and metal combinations to screen. The reaction condition scope is more or less well defined from the start, and you can see immediately if it works or it doesn't work. In biocatalysis, very often, you have big libraries to screen, or you really don't have an enzyme that works well enough, and you need to kind of design one, right? And maybe we can get there in how you run your research. So it needs a set of competencies, which is not necessarily the bread and butter for the typical synthetic organic chemist.
Yeah, definitely. I think that's probably one of the biggest challenges that I think has kind of impeded broader adoption of the method in the field. That I think a lot of synthetic organic chemists feel like you have to be in the club, or you have to have some sort of training or expertise in the area to be able to carry out the transformations routinely. I think to some extent this is true, because ideally you would want something that's that you can just buy, and it comes in a bottle, and you can simply just scoop some out, dump it into your reaction mixture, and let it run, and like a traditional organometallic catalyst and so on. And this is not the case right now with enzymes. A lot of the enzymes that we use are not commercially available, so that's definitely a barrier of entry. And there are possible ways to address this. I think that there are companies that are now kind of crowdsourcing the work, if you will, so that in the sense that they help academic labs commercialize their enzymes so that other people can use them. I think one company that comes to mind is Prosomic. It's a UK-based company. So hopefully, it's definitely a step towards the right direction. It's just that it might take time.
Yes, yeah, I understand. How do you approach it in your research? Because you're a chemist by training. How do you integrate enzymes in your workflows? Do you actually use existing libraries that you get from collaborators, or do you actually have a biocatalysis part of the lab and you actually do
we do. Yeah,
evolutions and things like that?
Yeah. In our case, we usually survey the literature to look for potentially useful enzymes that we can use in our work. And once we identify enzyme candidates, we would get their genes synthesized by an external vendor, and then we'll do the testing, the enzyme expression, and so on in-house. So we have the expertise for that. And then from there, then we can start thinking about how we can use the enzymes, for example, by first looking into the type of substrates that they can accept, and then that becomes a starting point into thinking about how we can apply them. So
you basically have a number of parallel sort of lines of research in your group, because you have the synthetic chemistry on one side,
Yep.
and then you have all the biology type of work, the biotechnology type of work, to be able to
Yeah.
to clone the genes and to express your enzymes and produce enough
Mhm.
to run the synthesis.
Yep.
And do you even run enzyme engineering work?
Just a little bit, not too extensively though. I think that's because a lot of our work focuses on the application side of things, especially in terms of making certain complex molecules. And in my opinion, if we still have to do extensive enzyme engineering, then it's like running two projects at the same time, because you have to make sure that all the downstream synthetic manipulations work, and then at the same time you have to do all the engineering effort. And the I mean, it's fine in theory, but the timeline might not match with what we need to do. At the end of the day, we need to publish the work, right?
Makes sense.
Yeah.
Makes sense. Makes sense. Uh, of course, a proper enzyme engineering project can last for several months, right?
Right,
and
exactly.
Not assuming that you have automation available and all these sort of things.
Yeah, I mean, if there is if there is the right motivation to do it, we definitely would pursue it.
So you are able to find good enough enzymes for your type of application, even without extensive engineering of the enzyme properties, which is kind of interesting, particularly because you run some very complex reactions, right, on very complex substrates.
Yep.
So feels like your starting points are somehow already quite there, which is kind of refreshing to me. Because in my experience, back in the days when you were starting from somewhere, yeah, you could see some activities. Typically, you had to do a lot of work before getting
right.
something which was really useful.
Yeah, I think this is. I think this is the advantage of being able to do it at around this time period, because I feel like a couple of decades ago the tools are not there to optimize these enzymes and also to identify potential enzyme candidates. But now with all the recent explosion in whole genome sequencing, we can now find a wealth of enzyme candidates to test, and at the same time, DNA synthesis has gotten a lot cheaper, so it's really easy now to get a hold of many enzyme candidates to test in parallel to figure out a good starting point for your work.
Do you want to describe some of your reactions, some of your work, something that you are particularly proud, or an example where the enzyme really did something that surprised you, that you maybe didn't even think that was possible in some way? Did you ever had that moment?
Yeah, I think that one particular work came to mind where we just did not expect some of the enzymes that we were using to work that well on a variety of substrates. So this work was published last year, and it had to do with the application of several enzymes from the potenzimycin biosynthesis. This pathway itself was discovered by my colleague at Scripps, Ben Shen, and we were able to leverage a couple of enzymes from that pathway to perform site-selective oxidations on a terpene scaffold. So the substrate that we were using initially is steviol, which is the aglycone of stevia, and stevia itself is the main component of the stevioside, which is the main component of the artificial sweetener stevia. So we observed that the two enzymes from that pathway work pretty well on steviol. But as we start modifying the structures of the substrate, we realized that they're actually quite promiscuous, and they work on a range of substrates, and that allows us to design a variety of synthesis to make many different natural products that belong to that family. And in one particular instance, we actually observed that one of the enzymes that we use actually exhibit a different site selectivity profile on one of the substrates, and that allows us to design a different type of sequence where we were able to use that hydroxyl group to affect a skeletal rearrangement of the scaffold to access completely different family of natural products. So that
this was completely unexpected, and it
yeah, we
originated
did
a
not.
kind of detour from the original plan.
In some ways, it's more in that we were exploring what the enzymes could do, and then we observed that this gives a different oxidation site, and that allows us to think about a completely new direction and expand the initial plan to include additional targets from that.
Makes sense. I am curious now in terms of how you organize your thinking flow and how you you plan your research work. So obviously, I suppose you have some sort of interest in terms of the synthetic target.
Mm-hmm.
You tend to you still focus on mostly natural compounds, don't you?
Yeah, for sure. Although I think we do want to start expanding to non-natural compounds or things that are not natural products, but we still want to focus in the pharmaceutical area, so compounds that or certain motifs that are present in a lot of medicines and so on.
Well, that makes sense because keeping a look at the potential application of your research is really helpful, isn't it? So going back to my original thinking process. So you have, on one hand, you have your targets, then you have your synthetic strategy because you want to synthesize a given compound. So you need a certain set of tools to get there, and you have all the synthesis design aspect of it. On the other hand, you might have the exploration work of what the different tools, or let's say working on your toolbox, right?
Yeah, for sure.
Which I suppose might be done in a different way, maybe even in a systematic way, or exploring new reactivities or new enzymes, what they could do for you, or whatever, new chemical approaches, new synthetic approaches. Are these type of work both happening in your team, and do they run in parallel, or do they ever converge? How do you organize? I'm just curious.
Yeah, I think that both directions are being pursued in parallel in my lab. So different lab members have different projects, and there's a small subset that are pursuing the development of new biocatalytic methods, as well as the discovery of new enzymes that catalyze new reactions well, I wouldn't say catalyze new reactions, but more like discovering new enzymes that exhibit different substrate and set selectivity profiles that we've never seen before. So we're pursuing both directions in parallel.
Okay, that makes sense. So once something is interesting from the more fundamental type of work, then you obviously implement it in whatever applications.
Right. Yeah. So we typically would.
Synthetic work there.
Yeah, I think that we want to first explore and discover new enzymes, and then once we see something interesting from this exploration work, then we can start thinking about how we can apply them, and also think about potential molecules to make, especially those that would be relevant for medicinal chemistry and drug discovery.
You make it sound as something obvious and easy, but
it's not.
When I think about it, yeah, it's certainly not, right? And how has it been for you? Starting from a pure synthetic chemistry background, getting to grips with enzyme discoveries and having to deal with genomes and all the biotechnology work. These are all disciplines apart in many ways.
Yeah.
And conjugating them together in a single sort of research line is incredibly complicated. So, becoming an expert of this all is really quite an achievement. So I am indeed impressed. How difficult has it been? I know you spent 1 year with a Nobel Prize, but or I don't know how long you've been in Francis Allen's group, but that is still a short time in my mind.
Yeah, I think so. In some ways, I think at least in my experience so far, the biggest challenge is to uh, be familiar with the technology. And so, in my postdoctoral work, I had to get adjusted to the terminology, the experiments. But I think once you're familiar with them, I think it becomes more smooth sailing. Uh, and I think it's the same also in my independent career. From what I've seen, a lot of my trainees, I think the most important thing is for them to see how things are being done. I think once, especially on the biology side, because typically people who join my lab are chemists, um, and I think once they get the hang of the experiment, they're not technically that challenging. So once they understand the workflow and the logic behind the experiment, and I think they can do it in a very comfortable fashion. And the other thing that I'm also grateful for is that during my training with Frances, so in her lab, people come from a variety of backgrounds. So she has people from bioengineering, chemical engineering, uh, chemists, biologists, even structural biologists, and I think that was uh, a very rewarding time for me because I get to learn the perspectives of many people from many different branches of science, and just trying to and learn how they approach problems, and also I was able to get myself familiarized with um, cutting edge techniques from different fields just by talking to these people. So I think that's how I got exposed to a broad range of science.
And biocatalysis is really multidisciplinary, right? So
Yeah, yeah.
It's remarkable when you can work at the interface between disciplines and join together the strengths of it. I'm impressed when you're saying it's not that difficult after all, right? If you understand the logic of the experiments,
Yeah.
You can do it. So a chemist can do biology, and the language seems to be at the beginning, at least, the major obstacle.
I think it's the initial barrier of entry.
But once you're there, biology is at the end of the day chemistry with a sort of other level of complexity, maybe some more uncontrolled parameters. But at the end of the day, it's still a series of reactions.
Yeah.
Exactly. It's an interesting and refreshing way of thinking about it. Some biologists might disagree with us, but let's think about something else.
Yeah, I guess we're approaching this from a very a general point of view.
Of course.
Yeah.
And it's a pragmatic point of view, right? Because
you
have an objective in mind. So what you're looking is very specific. You want something able to do the reaction you're looking for.
Yeah.
And that's. So I am always interested in understanding the drive, right? Because your work is extremely interesting, and if you love chemistry like I do, there's a universe just behind your papers, and you can see the thinking process, and there's a lot of creativity there. You obviously need to have the right training to catch the most of it, and I'm probably not completely up to it, but it is indeed fascinating. What I am interested in, it's always difficult to catch from the work itself, is what is really your interest. So are you driven by the problem, the fundamental understanding or the challenge of the synthesis, or are you driven about what this could mean in the future in terms of applications?
I think I started my career with probably my main motivation when I started my independent career was to develop strategies to make complex molecules by incorporating all these tools from the enzyme world. And I think I'm still interested in pursuing that direction in the future. At the end of the day, I like to make molecules, and I think that there's something about looking for the most efficient way to build things. Maybe it's part of our human nature just to look for the most efficient ways to build things. I mean, we see it everywhere around us, like right, like how we build stuff. But as I go along with my career, I think I also felt like I've become more and more interested in the application side of things in terms of how we can leverage some of these tools to make things that are useful for the society, and also even enable some biological discovery. And I think that that's nice. That's one thing that's nice about being at Scripps, is that we're surrounded by a lot of biologists and chemical biologists who are interested in important biological problems, and I think that gives me a lot of exposure to important problems in that area that await chemical tools to solve. And I feel like I've become more and more interested in thinking in that direction. So,
it makes sense, right?
Mhm.
You know, it's very natural to start from the intellectual challenge,
right? Yeah.
and then as you see that you are up to the task, and you can meet the challenge, you know, you can beat the challenge. You know, what's next? How can you look beyond? It is a maturation, so it's a growing kind of thing. It's a process. It's
Right.
beautiful. It's beautiful the way you describe it. You mentioned before the you know, application into pharmaceutical compounds of pharmaceutical interest.
Yeah.
So I guess your current interest in terms of applications or kind of problems that this could solve are in this direction. Do you have anything more specific in mind or target, at least for the future?
Yeah, there's an ongoing work in my lab where we're trying to explore the medicinal chemistry of a particular molecule that we made last year. So this molecule is a very potent inhibitor of the 20th proteasome, and there has been a lot of work in the area where people show that if you inhibit the 20th proteasome, this would sorry, let me backtrack.
Sure.
It's been shown that inhibition of 20th proteasome can give rise to anti-cancer properties, and there's a couple drugs in the market for the treatment of multiple myeloma that acts through this mechanism of action. And based on what we discovered with our with this particular molecule that we made, so it's called sipafungin, we became interested in exploring the medicinal chemistry of the molecule. We've made a bunch of analogs of the molecules, and we actually found one or two that are significantly more potent than the parent compound. So that's probably the most current work that's relevant in this area. So we're currently looking into pursuing in vivo studies with some of these compounds, and hopefully that something might work out.
Oh, that is exciting.
Yeah, it's an exciting new direction that I never imagined we would be doing when I first starting out.
And all is done with, you know, some remarkably creative chemistry, right? With new methods and ways that people haven't thought about before, which is amazing. Hans, it's been fascinating, and as we get to the end of our chat, there's always a question that I use to close the interviews.
Mm-hmm.
So which is an obvious one for people like yourself. You're still very young, but at the same time accomplished, right? you're a bright
Thanks.
star in the chemical community. And you have this innovation in your thinking, and you certainly have a bright future ahead of you. But
Thanks.
You're sort of already well advanced in your path, and so if you had to stop and look back, what would be the piece of advice that you would pass on to someone younger who is just starting?
I would say don't be afraid to branch out to new fields. I feel like going forward, science is just going to become more and more interdisciplinary. So I think there will be a distinct advantage of knowing how to approach problems from many different angles. So I think that I would definitely encourage people to expand their horizon and try to learn new areas if they have the time.
I cannot agree more. And you know what, Hans? I find very often that scientists can be extremely conservative, which seems counterintuitive, right?
Mm-hmm.
But once you have your areas of expertise and you like to stay in your comfort zone, I think this is human.
Yeah,
but,
for sure.
But your suggestion is your advice is absolutely agreeable. So it's a great one. So thank you very much.
Oh, thank you.
It's been great speaking to you. Congratulations for your talented 12, and for all the great work, and best of luck for the future.
Thank you. Thanks a lot, Paolo. This has been great.